So in all the previous sensors that we have been discussing, especially on this chemical sensor chapter, one of the important parameter that the designer or the user needs to pay attention to is the specificity and selectivity. Okay, and it means that, for example, when you design an ion selective field effect transistor, for example, you want to have a membrane that covers the electrode that will only pass through specific chemical ions which you are interested to know the quantity or the existence. You don't want to measure other things. You don't want to measure other chemical species within the sample. So that is the selectivity or sensitivity. Later in the biosensor chapter, also we will the sensitivity and the selectivity and specificity is also important, because normally people would like to design a sensor, a biochemical sensor that is specific towards specific analyte, specific biomolecule to be detected. Let's say, for example, COVID-19. It is caused by SARS-CoV-2 virus, and the PCR has to be able, for example, if we use PCR or we use other molecular diagnostic techniques like LAMP or CRISPR, or CRISPR, then the PCR has to be designed with primers that could identify and detect SARS-CoV-2 virus specifically, and does not detect any other beta coronavirus, for example, flu virus, influenza virus, or MERV virus. Yeah. So this is specificity and selectivity is usually important in biochemical sensor, biosensor, or also chemical sensor. Similarly with gas sensor, which we discussed last week, selectivity is usually a problem because, for example, tin oxide sensor, they can be sensitive towards various chemical gases, and they are sensitive towards carbon monoxide. They are also sensitive towards carbon dioxide, towards nitrogen oxide, maybe, although with different sensitivity range. Then, to increase the sensitivity, the researcher usually will add certain doping with doping, or will operate the sensor at certain temperature, at certain operating temperature, so that it will, based on that, then the the selectivity will change. Other method includes also to have a mixture of the ingredient, so not doping, but instead a compound of the oxide material. So not only tin oxide, but tin oxide combined with copper oxide, for example. And many different ways to increase the selectivity towards certain chemical species. Of course, it is advantageous in one way that we know what we are measuring with high selectivity towards certain chemical or biochemical species, then we know what we are measuring. We know that our sensor will be selective only to that particular analyte of interest. However, the disadvantage of having very selective sensor is that the development cycle is quite long. It is quite big. You need some time before you can discover or invent a sensor which is selective towards certain only a specific chemical species. Then, as the development time is also takes quite long, it will also mean that the development cost is quite expensive. Yeah. You need to do optimization. For example, in the case of gas sensor, you need to try different dopant, and you need to mix with different other semiconductor metal oxide with the tin oxide in order to test the selectivity. You test it with different various gas species. Yeah. You can try to Google old gas sensor papers, and they are usually like that, and they have the selectivity experiment, and it takes some time. It's not that straightforward. So it is costly. It takes a long period of time to have an optimized gas sensor or chemical sensor for particular chemical species. And one way to solve this, which even now is getting increasing again, and it is actually invented quite a long time ago. But at that time, the computation power is not that good. Just several days ago, I met with Dr. Kuat Triana from UGM, Universitas Gadjah Mada, the person who invented GNOS, electronic nose that can be used to detect or diagnose COVID-19 from the breath of a subject. Yeah. So he was developing actually electronic nose for quite a long time, even when he was a master degree student, maybe many years ago. And he told me that that time it was not that easy because computational power was not that good. Was not that good computation. He was using 486 processor. Maybe you don't know, you were not born yet, all of you. It was in the 1990s. Yeah, with the 486 computer from Intel. The processing power is very low compared to today's computer. And that time, actually, the theory has been invented. The theory that you have a certain array, an array of sensor, yeah, 9, 8, 4, whatever, 16. And each of the sensor has sensitivity and selectivity, different sensitivity and different selectivity towards certain gases. And they do not need to be very selective toward particular gases. Okay. And then the response of each of the sensor is recorded and the pattern is being recognized to recognize whether the gas contains certain gas species. Okay. And this is using a pattern recognition theory, which can be derived from statistics analysis, like principal component analysis, linear discrimination analysis, etc., or the recently increasingly used machine learning algorithm, which is related to artificial intelligence (AI). Why at that time it was not that prevalent, the use of AI or machine learning algorithm? Because as I mentioned, the learning period, the learning period of machine learning takes days, if not months, in order that the sensor array can have convergence in recognizing the correct chemical, the correct pattern of the chemical gas. Because at that time, computing power is not that strong. But now, within several minutes, several hours, even several seconds, when you have enough computing power, you can have your computer recognizing the pattern coming from the array of the sensor. So this is now becoming more prevalent, becoming more increasingly being researched. So you do not need to have a specific and selective sensor to detect a particular gas. Secondly, in real-world application, the sample may contain tens or hundreds of even thousands of gas species. For example, human breath. Maybe the main constituent of human breath when you exhale out will be carbon dioxide and maybe some remaining oxygen and of course nitrogen. But we want to detect the other metabolites coming from gases, usually in the form of volatile organic compound, VOC. These are small chemicals which usually becomes gas in gas form. And this is metabolite coming from the metabolism of the cells. And whether the person is healthy or sick, then the exhaled gas will be different. Okay? And this is becoming one of the important body gas, body fluid, not fluid, fluid also, but not liquid, body fluid that become one of the topic for recent research to be able to recognize the content of the breath. Previously, if you want to know the content of the gas, we have to use expensive instrument like GC, and GC and MS. GC is gas chromatography. Yeah, GC, gas chromatography, MS, and usually it is combined with MS, mass spectrometry. So with GC and MS, then the sample is being flown into a capillary tube, and then it will create peak with a certain mass, and the peak will form certain pattern, and then we can know the content of the gas within our breath. But the problem is with GCMS, the instrument is bulky and it requires special setup. It's not easy, it's not cheap. It requires special technician to handle the instrument. It's basically similar like the HPLC, which you can find in our lab, although that in HPLC it's used for liquid sample, while GC is used for gas sample. Okay? So an electronic nose can offer a low-cost alternative to of sensor to detect and recognize the patterns of gases coming from the breath. Yeah, and you do not need to every time rebuild it from scratch, like with the selective gas sensor. Yeah, with gas sensor, if you want to detect NO only, you modify, you optimize the tin oxide sensor so that it has high sensitivity only towards NO and low sensitivity towards other gases. It is still okay when the gases are usually atmospheric gas, yeah, NO, CO, SO, SO2, CO2, oxygen, etc. The problem arises when you want to detect, as I mentioned, the human metabolic gases. It contains maybe thousands of species, volatile organic compounds. How can we design specific sensor for each chemical or gas species? It is just not economical, not feasible. The only solution is to use an intelligent chemical sensor array. We have an array of uh, sensors which they do not need to be selective to specific species. They can be selective, but not very high selectivity. It's okay. Then, but as array, as a group, then we can recognize the pattern coming from the gas. And this is basically we learn from the principle of our own nose. Our nose, yeah, does not have as a system that is not selective towards a specific gas. Instead, it has these millions, or at least hundreds of thousands or thousands of olfactory neurons. Yeah, olfactory neurons. That is, each of the neuron maybe has a specific configuration of receptor in its membrane and selective towards particular gases. As a group, then they can detect different aroma or smell or gas, which we then will be recognized by our brain. So then we can learn, oh, this is the smell of rose flower. This is the smell from orchard. This is the smell of poop, for example, etc., etc. Right? Through our learning from our children period until now. So we learn we are being taught by our parents or by our friend, oh, this is the smell of perfume, this is the smell of what, etc. And then we recognize that smell. So if you can imagine how many smells types are there in the world, of course, many different kind of smell with different composition. Smell of flower, smell of whatever. Okay, so and this cannot be done using conventional chemical gas sensor. We have to do it using intelligent chemical sensor array. So the intelligent chemical sensor generally include a sensor array and the pattern recognition function. This is what makes it different with conventional sensor. In conventional sensor, you just need the sensor element, the sensor material, and maybe some electronics. But in intelligent chemical sensor array, you have also the software, the algorithm to recognize the pattern coming out from the sensor array. At present, the electronic or artificial nose, e-nose, and electronic or artificial tongue, e-tongue, have achieved great development. E-nose is an instrument comprising of a sampling system. A sampling system is the one that takes the breath sample or the gas sample. Yeah, if you look at how genome is being done from video, or maybe you yourself have gone through using train. You usually will have to go undergo a genome test. So you have to sample your breath first using plastic bag, a plastic bag. You blow inside, and then that gas you put into the machine. And the machine has this array of chemical gas sensor with differing selectivity. So each has different selectivity, and then it has also a built-in computer with an appropriate pattern classification algorithm capable of qualitative and or quantitative analysis, of simple and or complex gases, vapors, and odors. In the case of, so it can be used to recognize various gas and samples. So the structure is basically resembling that of our biological nose. The sampling system consists of course, our nose and the holes. It has also some hairs inside it to filter the dirt, etc. It has also temperature, humidity control, because it connected with our human body temperature control system. Then it has the sensor array here at the olfactory bulb. Sensor array that contains 10 million cells, 10 billion, 10 to the power of 7 cells of about 100 different receptor types. So even until now, the best electronic nose, human-made electronic nose, does not have so many cells like this one. Then it has this pattern classifier in our brain. It's a real neural network, which will then try to determine the pattern coming out from the signal from the olfactory bulb. Okay, so, and the eNose is made similarly. So it has this sampling system. In the case of genome, it is a plastic bag. Then you have inside an array of sensor, and you will, from this array of sensor, you will capture an array of signals. So not only single signal. Conventional sensor, in conventional sensor, you will obtain a single signal as a function of time. In array of sensor, you have an array of signals. Then this array of signals are being put into a computer with pattern classifier or pattern recognition, which will then recognize the pattern using a machine learning algorithm or statistical algorithm. There are still fundamental differences in both the instrumentation and software of human nose and electronic nose. So this table lists the difference. For example, in human nose, you have 10 million receptor, while electronic nose until now maybe maximum 100 chemical sensor manually replaced. In human nose, it is self-generated. Human nose can have 10 to 100 selectivity classes, while electronic nose can have 5 to 100 selectivity patterns. Then initial reduction number of signal from 1000 to 1, to reduce the number of the signal. However, this is something which maybe the electronic nose cannot yet mimic this performance. Human nose is very adaptive. It can, for example, it can be used to learn new smell. Can learn new type of smell. And the key is by the adaptation in our brain. Electronic nose, on the other hand, so far, it is maybe possible with more and more development, but for now, it is not that obvious, because when, for example, genomes, it has been trained, for example, to recognize SARS-CoV-2 virus existence in the breath, in the respiratory system. But let's say, for example, can we adapt it so that it can also recognize other coronavirus? It may be possible, but it may require some training. Human nose saturates. Saturates means that When it fails, it is basically the characteristic of our neuron. So if you remember the early chapters in our subject, sensor and measurement, yeah, the characteristic of neuron is that after a while, then the signal, the output signal from a neuron will be constant. It doesn't give any signal anymore because it saturates. Like when I mentioned the time in our skin, when we touch, we feel the touching at the first place, yeah, at the first place. But after a while, we just consider this as the common norm, and then we forgot that actually we are touching. Similar like when people put like this a cloth, when they wear the clothes, put on the clothes, they feel the touching. But after a while, they forgot that a cloth is actually unwrapping. So this is saturation in sensor signal. While in electronic nose, it is usually persistent. It will give a constant signal. In human nose, signal treatment is in real time. While in electronic nose, this can be done with G-nose, for example. It is done in real time. It depends on the pattern recognition hardware. In the past, it may not be so because of the low sampling rate of the system in that time, 20 years ago. But now, computers are very fast, microprocessors are very fast, and the analog digital converter is also very fast. In human nose, it can identify a large number of odors, yeah, at the same time. Yeah. But electronic nose, you have to train for each application. As I mentioned, for the genome, it is been trained to detect SARS-CoV-2 virus in the human blood. Can it be detected to other diseases? Yes, it may be possible, but you need to retrain them. You need to retrain the device for each of the uh, disease. Okay? Human nose cannot detect simple molecules like small molecules, oxygen, nitrogen, CO, etc. Electronic nose can. So human nose usually is used to detect volatile organic compound, some compound having larger than this smaller gas. So human nose detects some specific molecules, and electronic nose not possible in general at very low concentration. And human nose is associated with sound, vision, experience, etc. It is connected with the other processing of our human sensor by the brain. It is possible with electronic nose to connect also with other sensory system, with what we call as multimodal sensory system, but not yet so prevalent, not yet so many people develop this kind of multi-sensor system. In humans, it can get infected. Yeah, of course, when you get flu and other respiratory disease, of course, then your humans get infected. As for example, with people who has COVID-19, they will have loss of smell. Yeah, loss of smell, taste. I forgot the name. In electronics, it can get poisoned. What does that mean? It can get poisoned, then yeah, by certain gases, which will disrupt or disturb or even destruct its sensing property. So that's why then eNose is composed two main components: the sensing system and pattern recognition system, capable of recognizing simple or complex odors, smells, yeah, and an individual sensor used for detection of particular substance like CO sensor is thus not eNose. This is what we have discussed last week, yeah, a particular detection of sensor, particle detection gas sensor, and that they are not eNose. Okay, the sensor system itself consists of sensor array. It is the reactive part of the instrument. When in contact with volatile compound, so the sensor will react, which means they experience change of electrical properties. Yeah, it depends on the sensor. You can have the semiconductor metal oxide sensor, which will then the resistance will change. You can have also quartz crystal microbalance, QCM, where the frequency will shift, yeah, and many other different kind of sensor. Each sensor is sensitive to all volatile molecules, but each in their specific way. Most eNoses use sensor array that react to volatile compounds on contact. The adsorption of volatile compounds on the sensor surface causes a physical change of the sensor. A specific response is recorded by the electronic interface, transforming the signal into a digital value. So this is the type of sensing elements which normally use here, metal oxide semiconductor, as I mentioned. So this is similar like the Taguchi gas sensor I explained last week. Yeah, usually they are each of the sensor specific. These are usually made from tin oxide, SnO2, can be mixed or doped with other material. Yeah, lead oxide, copper oxide, etc. Conducting polymer, yeah, like polyaniline, what else? PVDF, polyvinyl difluoroethylene, quartz crystal microbalance (QCM), which will, upon absorption of gas, the mass will change, and then the frequency of the resonance frequency also will shift. Surface acoustic wave, similar like QCM, but only the gas interact with the surface of the sensor, the adsorption. It can also be a field effect transistor. Okay. So the gas sensor array, so this is an example made by the Zhejiang University. The author of this book is from Zhejiang University. This is electronic nose made from TGS, ya, Taguchi gas sensor metal oxide semiconductor. Here, TGS, so for example, the type 813 is sensitive for hydrogen, isobutene, ethanol, methane, and carbon monoxide with 510,000 ppm detection range. 822 TGS is sensitive to these gases, and this is the range, etc., etc. And there is also two other sensor from MQ, and metal oxide gas sensor. This is a different brand, I think, which is sensitive towards other. So you see here they have cross sensitivity. Basically, different sensors have similar sensitivity or selectivity towards some of the gases, but they are also selective towards other gases specific to that particular sensor. The detection range also may differ between these sensors. Okay. So low concentration gas mixture were prepared, employing the possible biomarkers in the lung cancer patient's breath. Okay, so this is an example of using this array of sensor to detect cancer in the patient's breath. So this is the response of each of the sensor as a function of time. Okay, so you see here after breathing out, then it will take out, and then this is the pattern coming out from 8 of these sensors. Then they also use SAW gas sensor, surface acoustic wave gas sensor, combined with a capillary separation technique. Okay, this is shown in this structure is shown in this figure. So first, the gas. This is the sampler basically. The sampler with using CG is capillary gas separation. The respiratory gas is enriched by an adsorption tube. This one, adsorption tube. The desorption happens in the inlet of the capillary at a high temperature. Yeah, this one has the temperature control oven and capillary column. So the gas is flown here. The VOC, the volatile organic compound, is carried into the capillary to be separated by the K gas. The CG is carrier gas. Okay, this is the carrier gas being mixed. And when the VOC, volatile organic compounds, come out from the capillary, coming out here, there will be a frequency change because the VOC can attach to the surface of the surface acoustic wave sensor independently with the SAW by reason of condensation. And then the recognition is done by using PCA, principal component analysis. This is a well-known technique in statistics, and image analysis for pattern recognition. One detect result of the mixed VOC of the sample by Ines is shown here in for this figure. This is the spectrogram. So each of at different time it will have a different shift, yeah, frequency shift, yeah, because it interacts with different part of the surface acoustic wave sensor array. So this is similar to GC technology, basically gas chromatography, because it uses this capillary separation technique. So it responds to have two components: the appearance time, here when does it appear, at what frequency does it appear, and the response intensity. So the capillary from GC technique, the peak time here when it appears represents the time of each component of that gas. used to pass through the capillary. So usually then the one that appear at the earlier time is the smaller gas, smaller VOC, volatile organic compound, and the one appear at the later time is the heavier gas. The appearance time of the peak can be used to determining material. Because the capillary separating technique is applied, some disturbance factor in the environment is separated in the peak time. No influence on the substance. So the SAW gas sensor combined with capillary separation technique can simulate a virtual sensor array containing hundreds of orthogonal, non-overlapping sensor, which can detect and distinguish hundreds of different kinds of gases. Yeah, this is the theory. So because they combine with chromatography-like technique. And the pattern recognition, after we obtain the pattern, the signals from different sensor array. Okay. So this is, for example, conducting the response from different sensor element in a conducting polymer sensor array. Okay. So the initial resistance, then when the odorant, the volatile organic compound is introduced, it will give different response, different. This is during the absorption, adsorption, and this is during the desorption. So from there, then you can draw the graph, generated from the raw data. For example, here A and B values taken from the average signal from 15 to 17 seconds in A. This is while B is values taken from 55 to 75 seconds. So from here in A, 15 to 75 seconds is during the adsorption. Then B is 55 to 75 seconds is during the desorption, this value, the range value here. Okay. And we see that the sensor has different sensitivity towards the odorant, towards the gas sample. And this can be put into principal component analysis like this. So upon the introduction of certain gas stimulus, or odorant, or vapor, or volatile organic compound, it will have this kind of pattern between A and B of this 6 sensor. So, as a chemical sensor system providing several variables, in this case is A and B, a multivariate distance measure is therefore more appropriate than a simple univariate distance measure. So because then you have the two variables A and B here measured at different point of time from each of the sensor. So then this can be calculated using euclidic distance, the length of the vector connecting 2 points in the variable space between the A and B here. So ED sigma equals to square root of sigma 1 to n XA minus XB. The response, XA is the response of the sensor number N produced by sample A. XB is the response of sensor number X, the response of sensor N with sample B. Basically, the distance between this response and this response being squared, and then sigma of all 1, 2, 3, 4, 5, 6. Okay, and then you take square root of it. This is the euclidic distance. However, this euclidic distance does not take the variation within classes into account. A more appropriate distance measure is called as Mahalanobis distance, the statistical distance. Here is the statistical distance because okay. The statistical distance is calculated as the ratio of the euclidic distance and the class variance in the direction of vector among class centers. I think for this one, Pak Fatoni will be the expert in statistical analysis. Direction of high variance within the classes will just give a low statistical distance. And we have here then, after we obtain this data and recognize the pattern, then we classify and reduce the dimension. So classification is the task of making capable of assigning observation into different classes, making a model capable of assigning observation. So usually it is combined with dimension reduction in variable space. So is it really, for example, rely on the 6 different sensor, or maybe it can be reduced into 4 or 3. So a multi-sensor system produces data of high dimensionality, so because then it depends on these many numbers of sensor, right? Characterizing each observation. However, for us, usually our observation is limited to only 3 dimensions simultaneously. Yeah, that is why usually it is wise to reduce the dimension first. Yeah, it is difficult because to visualize more than 3 dimensions simultaneously. So the variable space is an essential concept in order to grasp the idea behind many data processing techniques. In the variable space, variables are seen as orthogonal basis vector, yeah, and the way to reduce the dimensionality is to find new dimension in the variable space, yeah, and use only the most influential direction as the new variable. So we reduce, for example, in the previous example from 6, we see which one is playing role. From there, then we can reduce the dimension. The basis change is made and dimensional reduction is performed. In a principal component analysis, a transformation projection in the variable space is made. And this can be looked at figure 4.51 like this. Yeah, this is a principal component analysis. This is the raw data in A. Then the first component, and then we see that it has contained 3 variables initially, but then the first principal component is the direction with the most of the variance in the data set. In B, we see that most variation occurs in upon variable 3, PC1. Then the low dimensional projection of the data, which is in the variable 1, can be used as a simple but good approximation of the data set. So this can be reduced from 3-dimensional data into 2-dimensional data. This is what we call as dimensionality reduction. When we can reduce the dimensionality of the data, then the recognition, the classification will be made simpler. And we can put, that it will be more guaranteed to have a convergent learning if we put the output into an artificial neural network, for example. Yeah. So as ANN, artificial network, is inspired by human brain. Usually it consists of nodes, yeah, and layers, and then usually the node represents a single neuron. But this is it is modeled as a software. Yeah? For example, model this we learn in actually semester 7 in the artificial intelligence class. So each of the node will contain the sum of each of the input from the previous layer multiplied by a certain weight. The weight is in the connection, in the synapse between the previous layer and that layer. Like this, and then it is being summed. It is put into a transfer function. When the transfer function is a simple step function, then when the sum is above certain threshold, then the neuron will fire. Will fire means will give output of 1, etc., etc. The output signal here usually will depend on how many classes you want to classify the signal. For example, in the case of COVID-19, yeah, it is just positive or negative. So you have only 2 classes or 2 output layer class. Okay. So consists of 3 interconnected layers of neurons. The computing neurons, the hidden and output layers, have a nonlinear transfer function. And it can be, there are many different algorithms of artificial neural network. The easiest one is sigmoid AAN, and you have a more complex artificial neural network. Okay, electronic tongue basically works with similar principle as electronic nose. However, because of time, let's for now, and we will continue in the next